to be more like an application talk, and I'll be introducing a lot of things. So first of all, I just want to know about you guys, like how many of you have any taken coursework or have a degree in biology in undergrad level or above? Okay, quite a few. And so how many of you have like a background in the physical sciences, engineering, computer science? So most of them, okay. So that's give me an idea of what to expect and what to do in this lecture. So what I will be doing is I'll be dividing this presentation into two different parts. So first of all, I will be introducing more about general concepts of biology and bioinformatics, what it's all about. And in the second part, we'll be diving into more core topic of today's, that's the applications of machine learning in different areas of biology and medicine. So let's start with the general introduction of molecular biology. So we all know that, like in our human body, each and every part of a human body is made up of some kind of cells. And these cells have, these are the functional unit of life, and they contain some genetic material. So this genetic material, which we know as DNA, that's the deoxyribonucleic acid. So it's a kind of a molecule that contains all the necessary instructions for life. So whatever you are doing right now, like you're breathing, there's a lot of biochemical processes going in your body. So all these instructions are controlled by this DNA. So this molecule essentially contains all the necessary instructions, or it can be also called as the molecule of life or software of life. So they contain the necessary instructions to conduct each and every aspect of the life processes. And these genetic material are generally present at the nucleus of the cell, and they're also present in the outside the nucleus, but majority of these genetic material are present within the nucleus. And these DNA molecule that I just talked about are condensed in the form of a chromosome. So I think you all must be familiar from your high school biology, and this chromosome is actually made up of a long stretch of DNA. So what is really DNA from a very chemical nature? So DNA is basically contains of it's a chemical, right? So it has a phosphate group, sugar group, and a nitrogen bases. Right? And it's a polymer of this nitrogen bases. So millions and billions of these nitrogen bases are stacked together to form a long chain of DNA. And this DNA takes the form of a double helical structure, as you can see here. And this DNA is made up of 4 different chemicals that we generally call as a nitrogen bases or nucleotides. So it's called the adenine, guanine, cytosine, and thymine. So we just abbreviate these molecules as A, G, C, and T. So all the life forms, from simplest organisms, say viruses, so bacteria, to cats, dogs, or even human beings, they're all made up of these 4 chemicals: adenine, guanine, cytosine, and thymine. So, and we abbreviate them as A, G, T, and C. So all these, the DNA from all basic life forms are made up of combination of these 4 different molecules. So these 4 molecules are stacked together in a different combinations to form a long chain of DNA. So you can imagine in human being, there are about 3 billion nucleotides. We call it as base pairs. 3 billion base pairs of these 4 molecules. So you can just imagine the number of combinations that can arise from these 4 different molecules, right? And so each of us have roughly about 3 billion of these nucleotides, but the combinations in each of us is unique. And that's what leads us to the variation or the uniqueness for each and every one of us. So maybe like, we all are human beings, so we all are Homo sapiens. So that means we do share a lot of combinations of these four molecules, but still we like each of us are different. Each of us are very unique. We have different facial structure. We have different, say, bodily shape, and different other aspects. So all these variations are a product of the different combinations of these four different molecules. So that is an essential concept. Okay? And so you can see here the thymine. So like thymine always pairs with the adenine. That is, T always pairs with A, and the guanine, that is the G, always pairs with the C. So so this is one particular strand of DNA, and the other is the other strand of DNA. So the two strands of DNA intertwine together in a form of a double helical structure. Okay, so everybody is clear till here, right? Okay, so as I was talking about, like, in a human being, there's 3 billion base pair. So in viruses, so the total genetic material that we call it as like a genome, that's the total genetic material, is about 170,000 base pair. So there are about 170,000 base pairs of these combinations of A, T's, C, and G's. And similarly in bacteria, it's about 4.6 million base pair. Fruit fly, 130 million base pairs of DNA, like genetic material, and even higher in plants. So in each and every different organisms, the total size of the genetic material really differs, and also the combinations are very unique in each of these species. As I just talked about, like, this DNA is present in the form of a chromosome, and the DNA can be divided into multiple segments, which is called as like a gene. So gene is actually a segment of DNA that codes for a particular protein, right? So gene is basically like, a combination of different A, T, C's, and G's, as I just talked about. And this gene leads to a particular protein. So each gene carries out some essential functions, and then function is carried out by a particular protein. So the main message what I want to deliver here is that these genes are the functional unit of the DNA, and they have a different each gene has a different combinations of these nucleotides, and they carried out some special functions. For example, the function of gene 1 may be different from the function of gene 2, and may be different from the function of gene 3. And they basically are just a different combination of A, T, C's, and G's. And in human being, there are about 20,000 different genes. So you can just imagine the sets of instructions that's carried out in all those 20,000 genes. To carry and each of those 20,000 genes performs, has a different function to perform. So this is an example of breast cancer-associated gene that's BRCA1 gene. So this gene is particularly associated with the risk of having breast cancer. So for information, Angelina Jolie, the famous Hollywood star, had some kind of changes in this particular gene, so that led her to decide, to just remove her breast so that she may not get the breast cancer in the future. So, like you can see here that this particular gene has a particular function that is which is very much it's associated with the risk of having breast cancer. So the sequence or the genes have different combinations of A, T, C's, and G's. So you can, from a computer science point of perspective, This is a very long stretch of text characters, or like a string, a very long string, right? And we can again, if we go around this link, so all this information is present in some kind of databases. So I just want to give you a rough idea of what a gene really looks like. So there are various databases that compiles all the information of the genes. So this is the same gene that I was talking about. So you can see here, this gene particularly starts from around this area and has like A, T, C's, and G's, but in different combination. So you can just imagine, there's like a long stretches of different combinations of A, T, C's, and G's, and it goes and goes and goes and on. So this is like, all these combinations of these nucleotide molecules. They code for some essential proteins, and they carries out, they carry instructions to carry out some specific function. So is it clear up till here? Okay. So as I just talked about, so this is about like the central dogma of molecular biology, and it explains how the information or the genetic information flows in the cell. So I just told you that all the instructions is carried out, is present in the DNA, and DNA contains genes, right? And these genes code for another molecule called RNA, that's the ribonucleic acid. And this is very similar to DNA, right? And this is single-stranded. And this RNA actually controls which genes turns on and which genes turns off. And then the RNA, in turn, produces protein. And protein carries out protein carries out a particular function. So the information flow is unidirectional from DNA to RNA to protein. And this is an essential concept in molecular biology, right? So, as I just said, this information present in DNA is transcribed in the form of RNA, which carries out the message, and also this message then turns into protein. It carries out a particular function. So all this DNA, RNA, and protein, you can just think as of different combinations of different molecules, and this is a long text of strings, which makes us and from say from a computer science point of view, we can look at this different combinations of A, T, C's, and G's in DNA and RNA, and also different combinations of these are called the amino acids, and also there are 20 different amino acids, and these amino acids have different combinations in different protein. So from a computer science point of view, it's a long text of strings, which makes it possible to decipher all the necessary functions of life. So back in 1985, the scientists proposed that why not to identify all the 3 billion base pairs of 3 billion nucleotides of human DNA and try to find out their order, or try to find out their sequence, in which order all these A, T, C's, and G's are present in the human being. And and then they conceive a project called the Human Genome Project to identify this particular sequence of in human being. And this is a very large-scale project, And this can be compared to some of the biggest projects in the history of humankind, like Manhattan Project, the atomic bomb project. It's similar in that scale. The Human Genome Project was launched in 1985, mostly with the leadership from the United States, and also with collaborative partners from different other countries, including UK, China, and many more. Before trying to find out all these nucleotide combinations of human DNA, the scientists also tried to identify the combinations of genetic material in different other life forms. For example, viruses, bacteria, worms, mice, and then humans. And the entire project, it started from around 1990s and ended up around 2003. And between this process, lots of progress being made. Right? Just prior to this project, like biologists used to only deal with one gene or two genes or a very handful of genes, and they did not require lots of computational power to analyze the data. So from 1990s, when they started sequencing the human genome, they were dealing with a massive amount of data. Right? So they were dealing with 20,000 genes, 3 billion base pairs, and so they were just going crazy. So they had no other way than to take help from computer scientists, and this really gave rise to the field of what's called the bioinformatics. But essentially, bioinformatics was introduced back from early 1960s, 1970s. There was lots of different classical algorithms already being proposed back in 1960s and 70s, but biologists was not very keen on using all those computer science algorithm. But from 1990s, they did not have any other way than to go and adopt computer science tools and to analyze their data. So this Human Genome Project really kicked all those development in the field of computer science to solve biological problems. And that's what essentially is the field of bioinformatics. And coming back to the Human Genome Project, the Human Genome Project was the first draft of Human Genome Project was completed around 2000, and the final draft, the final version of Human Genome Project, that is all the scientists found out all the necessary, say, the order of this A, T, Cs, and Gs in the human genome, finally in 2003. So this picture is from the announcement of the first draft in the 2000, where the President Bill Clinton himself announced that human beings were able to identify all the necessary instructions and necessary the combinations of these nucleotides that makes up a human being. And so you can check out these very awesome documentaries on DNA, on the discovery of DNA structure, and also about the Human Genome Project and how computer science really helped save the Human Genome Project. So at your convenience, you can go home and look at this. There are links available, and you can watch this documentary. And also, I would recommend you to read about this very awesome article about how Pearl saved the human genome. So as I just was talking about, biologists were dealing with a massive amount of data, and they did not know how to go about analyzing this data. So they went to some computer scientists. Actually, there were a couple of people who were making money in the video game industries, and they were the ones who actually saved the Human Genome Project. They wrote some fantastic algorithms to do some special stuff, and that really saved the Human Genome Project. And this Human Genome Project was, estimated to be completed in about, say, 30 years. But, with the help of computer power, that really helped shrink the time of Human Genome Project to about half, and also reduce the cost tremendously. So I'll just briefly give an overview of how the DNA sequencing is performed. So DNA sequencing is done to identify the combinations of A, T, Cs, and Gs, right? So for this, we first of all have a DNA molecule, this DNA molecule which has a 3 billion base pair, but the combination is unknown to us, right? So experimentally, what we do is we fragment the entire DNA molecule into very short chunks, right? And then each of this fragment is then copied. We make several copies of each of those fragments, and then we feed into an instrument called the DNA sequencer. And each of these fragments, it's processed, and then there is a very very complex chemistry involved that I will not be talking about. And essentially, the output of this is the combinations of A, T, Cs, and Gs in each of those fragments, right? So now you can just imagine the number of fragments that is obtained, and it's a very challenging task to put back all these short segments together back again in the form of DNA. So everybody is clear or confused? If anyone is confused, just raise hand and ask questions.
Sorry, so did you say that there are always certain pairs, like G and C and A and G?
Yeah.
But then I see something like there are double T's.
Yeah, exactly. As I just mentioned, there are 4 essential nucleotides that makes up entire all life forms, A, T, C, and G. But their combination can be different, and there could be repetitions too. Okay? So if I just go back to this particular BRCA1 gene, so you can see here there's a different combination of A, T, G, and Cs, but there are also some repetition of G, 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 G. So in many parts there could be also repetitions, so different combinations. So you can just imagine the number of combinations that's possible. So is that clear? So the combination, of course, it really depends on lots of stuff. But what do you mean by really valid or not?
I can take the GGC and do a combination, then I can have one sequence, right?
Yes.
So how can I say that sequence is related to the GI?
Okay. So of course, the combination, there could be any kind of combinations actually. Different genes have different combinations. So of course, even if you randomly shuffle all this ATC gene combination, and you can just construct a massive, you can make a combination of 30 different nucleotides at 30 different positions, right? And any kind of random combinations, if you if you give to a particular chemist, he can construct the DNA from those combinations. So any combination is what we call is valid, but the problem is that not every combination can give rise to the necessary instructions. So that instructions is very, very unique, and it has some grammar involved. So there are some syntax. There's we have to go deeper into molecular biology to understand that. But what I want to say that of course chemically you can have any kind of combination sequence. You can get a DNA, but that DNA may not generate the necessary RNA and protein to construct a function. To produce an RNA, there should be like a specific, it should follow some specific grammar involved, and only then it will be like very active or very functional. Is that clear with everybody? Yeah. So now, as I said, when we do the DNA sequencing, we, yeah.
I have one question. How were they able to predict, estimate the numbers of base pairs?
So actually, they sequence. So like the human, the Human Genome Project, the essential task of Human Genome Project was to sequence the entire human genome, the entire DNA of human, right? And then when they sequence, they can identify how many nucleotides, how many base pairs make up the entire DNA, and then they can just count it. Is that clear or not? So say you have one piece of DNA, and then you perform DNA sequencing. So you can know at each and every position what is which nucleotide is present over in that. And then finally you can count the number of the nucleotides present in the human DNA. And that give in human being that was estimated to be about 3 billion base pairs. Right? Is that clear? Okay, so so after DNA sequencing, so after we know the sequence of the DNA, but now the problem here is that we only know the sequence, the sequence of these short fragments, right? So now we have to go back and assemble all these short segments back into the human genome so that we can have a full stacked layer of the entire string of DNA. So so all these, the arrows that I'm showing here, those are the fragments of DNA that were sequenced. So all the sequences known for the small chunks of DNA. And then from the Human Genome Project, we know the entire stretch of DNA sequencing, all the nucleotides. And now the task is to just go back and assemble the individual short fragments into the human reference DNA, and then we can do and stitch together all the nucleotides. Yeah.
So previous slide you were saying that several fragments are being made. Are all fragments copied the same number of times, or
A different number of times.
and
Yeah.
why do we need several copies for DNA sequencing? What is the advantage?
Yeah, so the advantage is precision. The advantage is about how if you sequence once, then you might sometimes you might get some errors, experimental errors,
Is there a
right?
trade-off? So technically I could just make several copies of every fragment and that'd be good? Or
Yeah, so of course, if you copy, if you just copy the fragments, then you will boost your confidence to read out all the order of the sequences, right? So if you just have one copy, it will still work, but your confidence would be low. And if you copy the fragment into multiple times, then you will be able to sequence multiple times. And then when we sequence multiple times, and if you get the same combination of nucleotides, then you have a boost of confidence that whatever you're doing is correct.
So by making copies, all you mean is copy pasting?
Yes, yes.
So that is an easy task, right? So you can just make as many number of copies you want so that your confidence is maximum.
Yes, exactly. So there's also some technical trade-offs, like you can altogether you can sequence some fixed amount of nucleotides at a time, and that's the money is also taken into account, like when you spend all those chemicals. So do you want to only focus on one particular segment of DNA and sequence that DNA with a very high confidence, or you want to look at the entire stretch of DNA, but you want to have some shallow regions of fragments for sequencing. So of course there is always a trade-off. Yeah. So so these stretches are the multiple copies of those DNA, and you can see some areas are overlapped. So in the overlapped region, you will have a very high degree of confidence that that whatever we are doing is correct. And sometimes, of course, there might be some discrepancies between one particular fragment and the other fragment, and sometimes we might have to neglect if there is some kind of errors. So is everybody clear up to here? Okay, so this is just what I was talking about. This is the reference human DNA, and these are all the reads that we obtain from the DNA sequencer. And this has A, T, 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 G, C, and this fragment has A, G, A, G, A, C, C, T, A, A, G. So these fragments, then we can align back into our target genome. And this problem is another problem, very essential problems in the field of bioinformatics that was first introduced around 1960s, 1970s, and it's called the sequence alignment problems. And it's just about aligning some stretches of strings into given we have a template sequence. So this is the sequence that we obtained from the Human Genome Project that is already known. And then, say, for example, we are trying to sequence DNA for, say, cancer patient. So all this DNA are from the cancer patient, and this is what is present in the database. So now we are trying to create or try to know the sequence of DNA in the cancer patients. So by doing DNA sequencing, we can know all these chunks of small fragments of sequences, and then we can align this back to the human reference genome from the Human Genome Project. We can align at which positions, and also you can see here, there's another read which share some fragment T, C, G, and here, and then we can stitch together these overlapping fragments together, and then we can go on stitching together to form a long chain of DNA. So this is what DNA sequencing is all about. I agree that this is a very brief overview. I have not clarified lots of details, which is very complicated to understand in a very short amount of time. But what I just wanted to deliver the message that with DNA sequencing we can know the order of the sequences in the human genome. So is this clear with everybody, or is there any questions? Yeah.
I had a question. So this whole 3 years project, we took a reference DNA sequence and managed to decompose it into known fragments.
Yeah.
So we can reconstruct the original using these fragments. But as you said, there are some parts which are specific, which can help us understand some functions of
So the whole, like the fragments which we have in 3 years, the fragments which we discovered are for that one reference DNA. So how will it be useful, or how can we apply them to other humans?
Yeah, you're very correct. Like in Human Genome Project, there was actually not only a single individual involved, but there were more than 20 or 30 different individuals' DNA was sequenced together, right? But of course, those 20 or 30 people came from different races, so their genome was sequenced for the first time. But you're correct that every individuals are different, and say every ethnic populations or racial populations are different. European populations might be different from Asian populations, and also might be different from the African populations. So individual and at the population level differences are there. But the human genome provides the basic, if you just forget about the differences, the basic nature of the DNA. And of course, there are different projects currently being carried out to identify the combinations in different races and different populations. So it's still in the at the stage of research, but when we want to look at the individual, there will be some variations, and those variations are taken into account with different the computer algorithms are there. So they will if there is some, let's say, chunks of sequence missing, then it will introduce some gaps within this the sequence for that particular individual. If there is a new stretch of sequence present as compared to the reference, then we will add the sequence. So it will take into account all those variations. But of course, each and every individual are different. So the reference human genome does not really represent the entire differences in the different population-wise or individual-wise. Any more questions? Okay. So in the Human Genome Project, it took about 15 years, and it took had a budget of over $3 billion to $5 billion. But after Human Genome Project, there was lots of progress being made in the in the field of chip manufacturing. New technologies were introduced, new computational tools were introduced, and that really helped shrink the cost of the human genome. And the cost of DNA sequencing is dropping rapidly, much rapidly than the estimated by the Moore's law. And at present, if you want to sequence a human DNA, it will cost about $1,000 US. And they're even trying to push this cost further. And with the decreasing amount of cost, you can sequence a lot more. So there is a tremendous growth of the data in very recent years. So this particular graph really shows how many genomes we have, the entire scientific community around the world have sequenced till date. So at present, we have sequenced just about under 1 million of human genomes. I'm talking about the complete human DNA. So for about 1 million individuals, we have already sequenced. And this, and the next 5 to 10 years, there will be some new technologies be introduced, and this will again push the growth of data. So what I wanted to tell you is that the field of biology is being inundated with massive amount of data coming out of each year, and biologists are overburdened with all these massive data that they have to deal with. So to tackle this problem, they had to go back to computer science, and it's all about bioinformatics, right? So bioinformatics, as I just mentioned, it's a combination of two words. That's biology and information technology. So bioinformatics essentially is a combination of statistics, computer science, and biology. So you can think of biology and also like computational biology. They are often referred to as like bioinformatics and computational biology. They can be thought as data science in biology. So you can find bioinformaticians or computational biologists in different roles. Some people work in the field of algorithm development, so they have skills skills in math, statistics, and computer science, and a very a good background of biology, and they develop different computer algorithms to solve different biological problems. And the other other bioinformaticians, they love to program, they love to code, so they work in the field domain of software development, and there's lots of job opportunities even in as a software developer in this field. And the other bioinformaticians, they like to use these tools and process all those DNA sequences coming out of DNA sequencer. So they implement different pipelines, different softwares, softwares in the field of bioinformatics, and they what they do is they take the raw data from the coming out of instruments. They they implement that particular pipeline, different softwares, and they produce some data ready to be analyzed by the biologist. And the other data analysis people, they have strong skills in biology and also very good skills in maths and computer science, and they love to analyze the data and from a biological perspective try to address different biological questions. So my point here is that there's an awesome amount of data present in biology, and computer scientists or bioinformaticians are recruited at different industries at different settings, and there's lots of career opportunities over there. So you can also browse through this particular portal. It's called the Omics Tools, and this contains lots of, this is a repository for different softwares in the field of bioinformatics. And if you want to just browse through this link, and you can get an idea of what other disciplines of bioinformatics, bioinformatics really work on. So this is one of the projects that I am working on. It's called the Pan-Cancer Analysis of Whole Genomes. So what we are doing is we are looking at 3,000 cancer patients. We are doing DNA sequencing, RNA sequencing, and looking at different aspects of those around 3,000 cancer patients. And we are trying to identify, try to address different questions. And of course, the main question is to identify the treatment for these individual cancer patients. And here, the size of the data is about 56 petabytes. So you can just imagine the amount of data present for this single project. I'm not, this is only a single project I'm talking about. And when this project started, 5 or 6 years ago, no single center, institute in the world was had the capacity to analyze the data at its own. So there was a consortium put, and there were like around 6 different supercomputers from different centers around the world. They agreed together to analyze this data. So all the 56 petabytes of data were spread in all the 6 different supercomputers around the world, and then they were able to process the data. So my point here is that bioinformatics is dealing with a big data problem, and that's where all the bioinformatics, where all the machine learning really plays a very great role. As I just said, the cost of sequencing is decreasing, the amount of data is increasing, and so is the cost of bioinformaticians. So like the investment that's put in to the field of bioinformatics is growing so much that it's growing on and on and on, and that is really triggering all the developments in the field of bioinformatics. All the they're coming up with computer scientists are coming up with very awesome algorithms, and so that to tackle different questions in biology. And of course, like the investment in bioinformatics is growing on tremendously, and there's a tremendous amount of career opportunities in this field. So with that, like I think, yeah. So one more thing is, many IT companies, like Google, Yahoo, IBM, Amazon, Microsoft, they are all investing a lot in this field, and each of these companies have their own wing that is dedicated towards the genomics and bioinformatics research. So even from a career perspective for a computer science students, like the field of bioinformatics is it's very exciting, it's very rewarding. So I think I would just invite everyone to explore this area. So with that, I think we can take a break for a few minutes. So, and like meanwhile, if you have any questions, you can just talk to me. Yeah. Stretches of combinations of A, T, C's, and G's, and they follow some specific grammar. Now, if there is any kind of changes in that particular grammar or in that particular chemical, for example, if in the where it was supposed to be A, there is now like a T. Like a different chemical is substituting that particular chemical. Or if there is a new chunk of DNA added within that gene, or if any nucleotide is deleted from that region, then it gives rise to some kind of mutations, right? And those mutations is errors in the DNA, and that is the causal factor for disease. And even for cancer, if there is any single change in that letter, then it can give rise to cancers, or other diseases, depending on the context. So those mutations sometimes might lead to some fatal diseases.
Let's say a healthy person is supposed to have identical gene for, let's say, breast cancer gene, right? That's what you're saying?
Yeah.
Okay.
Okay, next question. Okay, while people think about the question, I'm going to, someone, just wait a second, yeah? I'm going to let the students in.
Algorithms and to step forward in this particular field, I would say.
Okay, one final question.
Hi, thank you for the talk so far, Raunak. My question revolves around you mentioned about these 6 or 7 supercomputers around the world that was needed a few years ago. How is the field evolving, and if researchers in the low-income countries around the world want to do some fundamental research in this field, how much do you think they are, when they face these difficulties of lack of computational infrastructure or even experimental biological materials that they need, and what directions do you think there are opportunities and challenges for low-income countries?
That's a very interesting question, and I think I like to highlight, what's happening in India. In India, even in India, they do not have a very sophisticated computational infrastructure. So the bioinformaticians within the India and the different universities, they have come together to form some kind of open source consortium. They call it one project is open source drug discovery. So all the students working in the field of computer science, bioinformatics, biology, they come together in a form of community, and they use their own computational resources that are available in their college or home, a simple laptop, and they form a consensus of what to do. That there are some instructor for individual projects, and those instructor give a guideline of what how to carry out a project, and then individual student goes back to in their own laptop, carries out their specific instructions that can be done through internet and in a simple laptop, and then together they form a collaborative projects, and and from following the concept of open source, they populate all this data together in some, say, for example, Google Spreadsheet, Google Docs to share information, and they compile different data together and process together. But of course, doing some very big projects requires a lot of collaborations from different other resourceful countries too. And I think even the lower resource countries should come together to tackle some important questions and work together. I think that is the way forward.
Okay, thank you. We need to press on.
Okay, now we'll come back to our main topic. That's the applications of machine learning in biology and medicine. So what I will be talking about is why machine learning is a good tool for biological studies, where in biology and medicine machine learning tools are being used, and when not to use machine learning. So first of all, I will just go back into the basics, and I think you have gone through a lot of lectures in the past few days. But what I'd like to especially highlight is two different cultures of machine learning. One is the black box approach, one is the open box approach. I think you have all very familiar with this. Now, if you have some kind of a data available that's input data that's X, and you have some measurable outcomes like Y, and you want to model that relation. And one way is to model is called a black box approach, where you try to produce some, for example, say neural networks, and try to infer the relation between X and Y, and try to generate a model and perform, say, different classification tasks and so on. The other approach is called the open box approach, where you have some prior knowledge of X, how how your input data is, and you try to infer some priors on it. You have some assumptions, and you can model this data using different statistical tools, different concepts like linear regression, logistic regression, or Cox modeling, and then you infer the relation between X and Y depending on some your prior assumption. So just to give you an analogy, say if you want to you're dealing with a classification problem of image of cat versus a dog. In the black box approach, you try to classify the cat versus dog, right? And then you try to identify the features that distinguishes cat versus dogs. Now the classification problem is done, and you have those features, but those features may not be important for the end users. So I'm not interested in what, makes a cat, and what makes a dog, and what differentiate cats versus dogs. In black box approach, we are mostly interested in the classification problem rather than looking at the features. But in the open box approach, we have some prior knowledge about how cat looked like or how dog looked like, and the feature that separates between cats and dogs are essential for the end users. So just to clarify this further, what I want to say is, okay, in the black box approach, say if we have we are dealing with a diagnostic problem. So we have, say, an X-ray image of a chest from a human body, and we want to detect tuberculosis within looking at that image. Now, now we can develop some kind of neural network model and then classify which X-rays or which individuals have tuberculosis and which does not have tuberculosis. But we are looking at the images. We are not interested in what distinguishes the individuals that have tuberculosis with those individuals which does not have tuberculosis. But you can correct me if I'm wrong. But in the open box approach, we are mostly concerned, especially in the field of biology, we are mostly concerned with the question why, right? So for example, if we have a problem why does some patients respond to a drug while other patients do not? So if you give a drug, a particular medicine to a patient, now some patient might the drug might cure that patient. But for some patients, the drug might not work. So from a biology perspective, we are mostly interested in why, the question why. Why does that drug work in some patients but does not work in the other patients? So in this case, we are interested in the learning about the features. Of course, the classification problem distinguishing those patients that respond to the drug and those patients that do not respond to the drug is essential. But also we are interested to know about the features, like what makes the drug work and not work in some patients. So the first area of the application in biology is the field of gene regulation. So here we are interested to know about how the genes are regulated and so on and so forth. So what I mean by the gene regulation is that, as I said, there are 20,000 genes in the human body and human DNA, and at different points, certain genes are turned on and certain genes are turned off. And even for, say, some disease condition, in a healthy individual, like say gene A might be turned on. Now, when he or she becomes diseased, that particular gene A might be turned off, or vice versa. So, in some other disease condition, in the healthy individual, the gene is usually turned off, but when the genes turn on, then it triggers the disease. So it's very essential to know which genes are turned on and which genes are turned off, depending on the context. So for that, so if we just focus at this point region, so this particular region is a gene, and there are certain other proteins which binds to this gene, which binds to the stretches of DNA, and this triggers some activation signaling to turn on the gene. So if these proteins, which are called as transcription factors, so if these transcription factor proteins are bound to the DNA, then it gives an activation signaling to turn on the gene. And if those proteins are not bound to the DNA, then the activation signal is not triggered, so the gene is not turned on. So the pattern, so also the pattern of sequences that I talked about, the combination of A, T, C's, and G's, are essential to know about where those transcription factor proteins bind to the DNA. Okay, so so there are different problems. So one of the problems is to know about the patterns of DNA where those transcription factor protein binds, and this might be to hunt those patterns, different machine learning tools are being used. So to just give you a basic idea of workflow and how machine learning workflow really is carried out in this field. So first we have the raw sequences of DNA, and then the data is pre-processed, that is clean to to clean out a lot of stuffs, as I just talked about, sequence alignment, sequence assembly, and all those stuff. It comes in the data pre-processing steps. So after data pre-processing steps the data is ready to be analyzed, and then we perform feature extraction. That is, we identify different patterns of the combination of A, T, C's, and G's in that particular region where the transcription factor might bind. And then we train our model, in certain system, to identify, let's say, if we have healthy versus disease condition, then we train our model, and then we evaluate our model how well it can distinguish between those healthy and healthy and diseased individuals, looking at the transcription factors. So this is basically how the machine learning workflow really works in this field. Of course, there are different other approaches. You can also do supervised learning as well as unsupervised learning to model these kinds of data, and also sometimes the raw input data or the sequences are often high dimensional, and they might be related to the phenotype, say the healthy versus disease condition, in a very complicated manner. So so you can learn about different concepts of molecular biology and different grammars within the individual gene, how the gene is regulated, and then when we try to look at those high-level features and how this gene is organized, and then it could well discriminate those phenotypes in consideration, or different class levels in consideration, and also taking those high-dimensional representation, you can produce some say deep neural networks to model your data. So this is another example is to know about where the RNA so if we look at from here, so when the transcription factor is bound and the signal to the gene is turned on. So what I mean by gene is turned on is that it produces, the DNA produces the RNA. So RNA is the message, right? So, and it, when RNA is produced, it further gives rise to the, proteins, and it carries out the function. So when a gene is turned on, it produces different messenger RNA. And it is essential to know, from which position the RNA is really generated, that is from where the message is really starts. So, and that position is called the transcription start site. And to learn about those transcription start site is a very essential problem. So for that, you can gather some chunks of sequences, and and you can train on the data from where the sequence is aligned to the transcription start site. So some of the transcription start sites are known from, say, experimental approaches. And then you can give a class level of some have transcription start sites, some are not centered around transcription start site, and then you can build a learning model and a predictive model to classify in any test set which sequence might be aligned to a transcription start site and which might not. So this is a very generalized model of gene regulation of how or what different aspects we can do. So the next essential thing is how to go from the sequence to some kind of numbers, right? To model your data. So when you have different stretches of sequence, you can count the position. So say these are each line or each row here is a different fragment of DNA that are aligned together, like different fragments of reads coming out of, say, DNA sequencers. And then for each column, you can count the number of A's. So say for the first column here, there are 6 A's. There are no C's, so there are 0. D's are 0, and there are 2 T's. And so on, for each column, you can count the number of A, C, and T, and G's. And then you can also do some kind of normalizations in this counts to generate some pseudo counts data. So depending on your prior assumption, you can assign some weights and do different things based on some prior assumptions, and then you can calculate the proportions or the frequencies of individual A, C's, T's, and G's. And this data is what you can feed in your model. So it's things clear till here? I'm just giving the general overview of what things are done in this field. No questions? Okay, so just moving on, another aspects in machine learning and biology is about trying to classify based on gene expression data. So as I said, genes are turned on and turned off, and those the activity of a gene can be measured using some instruments, like even using the DNA sequencer, and then the activity of a gene can be quantified based on which genes are turned on. So if a gene is turned on, it will receive a very high number, and if a gene is turned off, then it will receive a low number. So so you can for each gene, each 20,000 genes, there could be assigned some kind of quantification value to model whether the gene is turned on and turned off. And now, say for example, if you want to look at a classification problem, healthy versus diseased person, or cancer state versus healthy state. So now you can do some classification problem and try to identify those genes which distinguishes those healthy individuals from the cancerous individuals. So, and if you try to find out those the features or the genes that distinguishes between the healthy and diseased individuals, those genes might be used for some development of some drugs, some medicines, some therapies. So that's why they are important. So this is a very classic machine learning problem, and what we can do is we can take a gene expression data. So all these the colors here. So each row is represented. Each row is a gene. All the 20,000 genes are represented here, And the columns, each columns here are, say, individual patients. And some patients are healthy. Let's say the patients represented with blue are the healthy individuals, and patients represented with the red color are the diseased individuals. Now you can split this data into a training set and a validation set. Now in the training set, you can just model your data, and you can validate in a validation set. And you can make a predictive model out of it, and using like an independent data set of this, like, same healthy versus diseased individuals, then you can validate your model and identify, like, how well did you do the classification. And you can also, the output, as an output, you can also identify those features or those genes that distinguishes between the healthy individuals and diseased individuals. Okay. So this is, like, based on the similar problem. This study, particularly, what they did was they looked at 1,000 different cancer patients, and they tried to look at all the DNA sequencing data and all different kinds of genomic data coming out of the body. And they tried to identify which, so each of these patients, each of those 1,000 patients were treated with different drugs. And they tried to identify which patients were responded to the drug, that is, which patients were sensitive to the drug, and which patients were resistant to the drug. So this is a very classic machine learning problem where you can model this data using some, say, logistic regression modeling, right? So you try to identify which individuals are responding or sensitive to the drug, and which patients are resistant to the drug. And not only that, but looking at, as I just said, looking at the gene expression data, you can identify the features or the genes which distinguishes between those responders or the sensitive versus the resistant cases. So which are the genes that are responsible for separation? Which genes are different between these two groups of patients? Now, after identifying those genes, those could be useful to study different biology, to work at for drug development and so on. And not only that, but you can identify based on, let's say, if patients which responded to the drug had a certain changes in the DNA or certain mutation in the DNA, but the patients that did not respond to the drug did not have that mutations. So they also try to identify different combinations, say for different, say, mutations within the different genes that distinguishes between those patients who responded to the drug and not responded to the drug. So this is just one of the results, and say each bar over here is a different drug. So say for example, yeah, so let's say the top example, this is one of the drug, Zemti, I can pronounce this name, zemticinamide. So so about, say, 50% or 60% of the patients which responded to the drug had a particular mutation in a gene called TP53. So, and the patients which did not respond to the drug, did not have these mutations at all. And so, they use some, machine learning models to identify all these mutations, all these genes that distinguishes between those patients that responded to the drug and not responded to the drug. So this is one particular application. This is another applications of machine learning in biology, and this is a very interesting and very controversial study where they tried to, for some individuals, what they did was they collected different kinds of data, like the data for age, height, weight, skin color, eye color, the structure of the face, and different, facial facial regions, as well as they took the voice, sampling of voice. And then also they took the blood sample and identify the DNA sequences, and they tried to correlate these features. And what they found was that they could actually predict, using machine learning models, they could predict which, from DNA, looking at the genetic material, they could predict what kind of facial features that particular individual has. So this means that looking at the DNA itself, you can have a lot of information about a person's individual's appearance, about the different ethnic features about race and ethnicity of individuals. So this is another interesting applications of machine learning in this field. So this is another interesting applications of machine learning, and this is from Google. And they developed a model called the Google Deep Variant. So this model really tackles the problem of identifying mutations, all the changes in the human DNA, in the DNA sequencing data. So the DNA sequencing data that comes out of the instruments has this kind of a particular format where they can look at individual nucleotide combinations at individual positions, and they can also identify different quality scores. So all these patterns shown in this particular figure represents quality, different, say, metadata associated with that particular position of nucleotide. And this is spitted out from the DNA sequencer. And what they did was they looked at this patterns of strings and treated it as an image, right? And then they tried to learn information from all the 3 billion base pairs of human DNA, and they tried to learn some patterns to look at what kind of changes that particular individual had as compared to the some reference human DNA. So yeah, and the output was whether that particular patient had some kind of mutation at that particular position or not. So so the concept here is that we can also treat these kind of textual information from strings, from string characters as patterns in an image, and then we can decipher these patterns using some machine learning models to solve some biological problems. So I talked about all those 20,000 genes, and each gene carries out individual functions. Now all these functions can be grouped together in a form of a tree, and that's what we call as gene ontology. So this is like a specific grammar followed by all the biologists to categorize individual genes into different functions. So the leaves of this tree is all the 20,000 genes, and they can be grouped into individual functions. Just for example, there's some hierarchy of cellular physiological processes which can divide it into cell death, cell cycle, and cell cycle has different steps and so on. All those functional categories can be grouped into form of a tree, And what interesting problem that people have been coming up is that now they are trying to associate those information from the DNA sequences to some kind of phenotypes. That if, when I talk about phenotypes, I'm talking about, say, it's whether a person is a diseased condition or a healthy condition, or some kind of, like, features that can be measured, some kind of traits that can be measured within an individual. So this particular this group from the University of California, San Diego, what they did was they model this particular tree, and they form a deep learning model using some kind of structure to it. So say, for example, if all the leaves here, so for example, if you consider this figure here, all the leaves in these trees are individual genes in the human genome. Now, if you do, like, if you just delete one particular gene in an individual and see its effect, following a structure, there will be some kind of, you can get some kind of relation of where the information is flowing, and what those changes in DNA is giving rise to the changes in the phenotype. For example, say if you have a cell in a laboratory setting, and then if you delete that particular gene, what will be an effect? If you delete another gene say B, then what will be an effect? And what different combinations of genes that you can take, and what, depending on what combinations, what is the results in that particular cell. So you can measure that. And then they try to construct some, based on the gene ontology tree, they constructed a deep learning model following some structures, information, and how the signaling flows within the neural nets. So is this clear? I'm sure, like, I'm just going very superficially, and there might be a lot of confusions, but I just wanted to give, like, a very brief overview of, like, what different applications are being done in this field. Any questions still now? Okay, I will just move on. So another interesting problem is what they have done is how to classify different cells based on their images, right? So in a cell, there might be lots of different types of cells. There might be, like, say, more than 20 or 30, say, for example, within a kidney tissue, there might be more than 10 to 15 different types of cells. And we are interested, biologists are interested to identify individual cells at a time, and they want to separate these cells. So based on, let's say, the images, this group, they constructed some deep neural networks to sort out the individual cell types. So based on, like, the machine vision, that's like, looking at the microscope and taking the image of individual cells, they were able, so this is like the microscope, and they photographed individual cells, and they represented the individual images in the form of some kind of, like, visual information. And then based on the morphology, based on how the cells look like within that images, they try to make some decisions on, like, which how to separate those individual cells. And now if you want to just extract, like, a special type of cells, and then this instrument will just spit out, like, the individual, that particular cell that you're interested in. So you can look at this figure. There are different kinds of cells, and each cells have different morphology. And so based on this morphology, this automated system sorts out, like, individual cells at a time. And then, like, those biologists can work on those different sorted cells into like different experiments. So, and this is another problem where they also look at the images from the individual tissues. So this image is from like a section of a tissue, like a section of a tissue from, let's say, from a cancer patient. And then they try to identify, or say they try to distinguish between different kinds of cancer looking at the tissue itself. So here they are looking at lung cancer tissues. So they try to classify, so there are two different types of lung cancer tissues, and two different types, major types of lung cancer, and that's what they are trying to distinguish. So based on the histopathology images, the tissue images, they separate the individual pixels, the information present within the individual pixels, into training set, validation set, and test set, right? So when after they split this data into different, like training set, validation set, and test set, they try to infer their model of how to distinguish between two different cancer types at a time. And then and also they also included different kinds of genomic information on top of it, and they try to infer some models of looking at the images of those tissues, of those cells, how they could distinguish between two different types of cancers, and also what kind of genes are different between those two types of individuals, two types of cancers. So the question that they're trying to address is looking at the images or looking at the genomic information, can they predict what kind of information they have, what kind of data, different kinds of cancers they have. So this is another interesting problem. So I will just introduce a last problem here, which is this is from my own lab. So what we are trying to do is we are trying to model a process in biology called metastasis. So cancer arises from a specific part of a body, and then when it advances, it progresses from its primary site, goes into the circulatory system, and it spreads out into different parts of different parts of the body. And this process is called metastasis, and metastases are very lethal. So in cancer patients which have advanced to the metastasis are likely to die in a very short amount of time. And so it is very critical to identify which patients might develop metastasis and which might not. So what the problem was that, we have a massive amount of data, but not all the data have a proper labeling of which patients had the metastasis and which patients did not have the metastasis. So we had the data back from early 80s and 90s, and most of the data was really useless to us. And we had say these are the old data which did not have the particular class labels of people which has metastasis and which are not. And these are the newer data which had the information about metastasis, which patients progressed to metastasis and which patients did not progress had metastasis. So we were interested to identify whether we can use this poorly annotated cohort as well as newly annotated cohort to develop some classifier. So what we did was we took this original input data, that is the data which did not have any kind of class label, and we used autoencoders to identify the most silent features that distinguish any kind of features, any kinds of labels, unknown labels present within the data. And then we reconstructed the input data. And then in the second step, what we did was, okay, now we also have another data which have information about which patients progressed to metastasis and which person did not. So we use this like all those features, and we further filtered out all those informative features which could which were informative for which patient had metastasis versus which patient did not have. And then we developed some classifying model using this signatures. Like these are all the names of the genes that distinguishes between those patients which progressed to metastasis and which patient did not progress to metastasis. And then we validated our model in different other data sets. So these are this is one of the interesting projects what we did, and this is called the deep genomic signatures. And I will not be talking, I'll just skip this part, and this might take time. And what I would like to conclude using that, there are some AI expert systems being used in the field of biology. One the case, an example is IBM Watson and Google DeepMind. So these systems are being used in different cancer centers around the world to identify, correct treatment for the different cancer patients. So one is the IBM Watson, and the other is Google DeepMind, which is being used in the UK. But the problem is that there was a lot of hype around 2015 and 2014, 2015 when it was introduced, and then very recently there has been a lot of negative news coming out in the media about if this IBM Watson is delivering its promises. Now how IBM Watson works is that it is a string, it's a query-based system, right? So the knowledge is present in the form of a curated literatures, right? And all these curated literatures are dumped in this IBM Watson system, and when we query some specific string, it gives out the answers for it. So for example, what treatment is available for patients having this particular mutations or this particular gene when is turned on and turned off, and then it gives out some answers. So the problem is that IBM Watson is not really giving a correct information for most of the patients, and that's because the inherent knowledge of from the literatures are not enough to make any kind of decisions. So, so what I believe is that in the future, when we have lots of information from different literatures compiled in the system, this IBM Watson technology will be more improved in the future. I think with that, what I like to say is that, okay, machine learning is really great to solve different biological problems, and of course, with the growing rise of biological data, machine learning can hunt for different hidden patterns within the, for diseases, but machine learning may not be useful for certain cases where the ground truth is not known. For example, in cancer, what causes the cancer is not known. So machine learning tools might not be very perfect when we try to address such questions where we lack the ground truth. And in the field of biology, there are lots of instances where we don't know about the ground truth. So what I would like to suggest is that, of course, machine learning is really great, but there are lots of other tools, lots of other algorithms out there beyond machine learning that you should be exploring and integrate with the machine learning methods. So I think with that, I would like to conclude my talk today, and like, we can take the questions over here or later.
So thank you, Raunak. So we're running very late, so maybe
The skills of an individual person, or say in this case, the skills of the pathologist or whoever, or the skill of the doctor or the surgeon is very essential. So if they give the right, the right part of the tumor, the right part of the tissue, then you will get the right information, and then you can do the diagnosis, right, or treatment decisions. But of course, like using they are humans. They can do their best of their ability, but they're also some parts of the tissues that might be undetected or unknown, which could be detected with the use of non-invasive tools like MRI, and they could complement in looking at different aspects of the tissue that might not be very well evident from the visual field of the surgeons. So of course, integrating these technologies is essential, and that will definitely help in aiding the treatment decisions. And there have been lots of work to integrate the different kinds of technologies.
Around some of the open data sets. For example, we have a lot of cancer data sets, repositories, and then we'll toy around and then use machine learning tools. Some of the questions will be, is there a silver bullet when it comes to analyzing those data sets, for example? And at the same time, if we are trying to produce that insight, can we say that this is a proof of concept? Because we need also to validate that with the medical practitioners.
Yes, of course. Most of those data present in the public repository, they are used to develop some different computational tools, machine learning tools, and of course, most of the information might already be known. And if you again rediscover the knowledge or the information using the computational tool, it will be a proof of concept. But you can later, after you develop your computational model, you can later use those models in a new patients that comes in the clinic. And of course, all the data present in the repository can be used for proof of concept. Given your question is right, given your data is right, and your model is correct.
okay, thank you.
Is the latent variable kind of thing. That is the genomic signature that you're learning.
Yes. So we are we are trying to learn this particular data does not have any kind of say class labels. So we are trying to learn if there is multiple class labels, what are the genes that distinguishes those class labels using autoencoders.
Okay.
So it can be any kind of unknown confounding factors present within the data sets.
I see. So whatever you're extracting as the features, are they interpretable by any means, or is this?
So each individual information is present for a particular gene. So we are hunting for genes that distinguishes between those unknown class labels.
So can you say that a particular dimension of what you see in the feature is corresponding to a particular gene? You can say that kind of.
Sorry, could you repeat that?
The whatever features that you learn, let's say it has a particular number of dimensions. Could you pinpoint that this dimension is representing a particular gene?
Yeah. So so what we do is we correlate those informations. Mm-hmm. Of course, it's not interpretable. It's a high-dimensional feature, right? And it's not really interpretable without correlation. So we correlate our original data with that particular high-dimensional representation, and we try to infer, okay, this might be the best gene that correlates to that features.